technical difficulties. I have been working on cancer projects for all my life, and this is my passion. And lately, I have been working on rapid detection of any DNA sequence. As I was working and developing the genetic services for Quest Diagnostics, as you know, the largest reference lab in the world. And I developed all the genetic services. And as I was making innovative findings, I felt that it is better to have my own company to accelerate the research. So about 10 years ago, I started this, and we developed the proprietary technologies. But the gist of all of it is everything that I have developed, I strongly believe in having 4 attributes compared to the gold standard. Any assay that I will bring has to be rapid, sensitive, cost-effective, but most importantly, amenable to point-of-care testing, so that it will be very, very useful in the clinic. So I guess Dr. Conero is probably going to be advancing the slides. So on this screen, you see the title, the assay title, Oncosure. And in the brochure or the invitation that you received, it says ultra-fast detection of all human cancers using liquid biopsy. And that's what I would like to share with you in the next few minutes, half an hour or so. And hopefully you will find it informative and educational. And next slide, please.
Just 1 second.
Okay, as I wanted to tell you that cancer is a genetic disease, no matter what you think of it. And as you know, there are 2 types of cancers. Solid tumors constitute the majority, 80%, and hematologic malignancies, about 15 to 20%. But there is a hallmark for every cancer. There is no exception. The normal chromosome number being 46, every cancer deviates from that number, whether it is lower than 46 or higher than 46. Most of the time it is higher, but sometimes it is lower. For example, phomochrobe renal cell carcinoma will have 23 chromosomes. But as clinicians, you all know that for the last few decades, you have been using clinical conditions, various imaging techniques, and serum markers such as the test, PSA, or whatever, to make the diagnosis. But in the case of the tissue, for solid tumors, you use the biopsy, and for hematologic malignancies, you use the bone marrow as the preferred specimen source, with the exception of CLLs, which we'll talk about. That's an exclusion criteria in this study. And it is an invasive procedure, as you know. Next slide, please. So for the last decade or so, liquid biopsy is a buzzword. I'm sure everyone in the audience have either used it or heard about it. And the things that are part of that procedures are circulating tumor cells, circulating free DNA, tumor DNA, and circulating free tumor RNA. So while these are mostly useful for prognostic purposes, in my professional judgment, they are of limited use for diagnostic purpose or screening purpose, and they are piecemeal assays, not comprehensive and holistic. Again, that's my individual opinion. And therefore, if you think about a screening assay for cancer, as you know, there are only 4 common screening tests: the breast, the lung, the colon, etc. But you all know the sensitivity and specificity issues related to those cancers. So therefore, an ideal screening test should be rapid, should be cost-effective, sensitive, and as I said, it should be user-friendly. It should be easy to use. As I will take you through the presentation, hopefully you will find the information that fits the bill as far as these attributes. And so what is the target for our assay? Oncosomes are the target. I will get into a little bit more detail about oncosomes. And also they mimic the parental tumor cell genome so that they can be used as surrogate markers. Next slide, please. So I'll just read it's a mouthful from this reference. Oncosomes are at present scientifically understood to be membrane-derived micro and nanoscale extracellular vesicles that are secreted by cancer cells and transfer oncogenic messages and protein complexes across cell borders. As I said, it's a mouthful. Let's discuss about them. Next slide, please. So this cartoon shows various extracellular vesicles that are coming out of a particular cell. It can be a normal cell or it can be a tumor cell, and we'll highlight the differences. Next slide. And oncosomes are part of these extracellular vesicles. So therefore, the working hypothesis is oncosomes mimic the parental tumor cells, and therefore they can be used to detect cancer and for screening purposes and monitor the treatment. But before we address that, you need to make sure that whatever we are isolating are actually oncosomes. As I just showed you in the previous slide, oncosomes are part of the extracellular vesicles. There are other bodies, and we'll concentrate on exosomes. I'm sure every one of you have heard the name exosome. And so first, we need to make sure what we are studying is oncosome. And second, we need to demonstrate that they do have all the characteristics of a tumor cell so that they can be used as a surrogate marker. Next slide, please. So here on the, let us start with the size. On the far left is an oncosome, and the middle is tumor cells. You can see the size difference. And then this cartoon is showing various extracellular vesicles. And next slide will show the blown up version of these extracellular vesicles. And you can see the green and other smaller circles are exosomes, and the blue are apoptotic bodies, and the red ones are microvesicles and large oncosomes. And at the bottom is given the size in the nanometers so that these large oncosomes are what we are going to be concentrating on. And I will show you the differences between them and the exosomes. Next slide, please. So here you will see the size difference as well as the gradient. And using the differences in the gradient, we can exploit that feature and clearly isolate the oncosomes away from the exosomes. Next slide, please. And that's a cartoon showing, I apologize, this is a little bit fuzzy, but I'll walk you through. If you take the peripheral blood and go through various centrifugation processes at various speeds, you can separate the cell debris, the dead cells, and the oncosomes from the exosomes. And we have gone through that process, and I will share the video through the website so that if we don't get to see it today, you will have that opportunity. So next slide, please. So here we have actually done that using the gradient. The blue line is showing where the actual tumor cells will end up, and the exosomes and the oncosomes will end up in the plasma, and then obviously the red cells are at the bottom. So it is the top layer that you will subject to differential centrifugation to separate the exosomes from the oncosomes. Next slide, please. So let's go through some of the features. As I said, exosomes are very small. And why are we concentrating exosomes? Because for the last 10 years or so, this field is about 10 to 12 years old. Exosomes have been used, or at least claimed to be useful for various applications in the cancer field. But most of the pharmaceutical industry has concentrated on exosomes in terms of the therapeutic purposes. So it is important to distinguish them from oncosomes. They are small, oncosomes are large. They are not visible under the light microscope. There is a reason for it. The next point will illustrate it. Whereas using the fluorescent microscope, since the DNA stain is for the chromosomes, we can see the oncosomes. And in fact, we are the first group in the world to show that these oncosomes have intact 24 chromosomes. By training, I'm a cytogeneticist. This is what I do for a living. I'm considered the chromosome doctor. So we have proven that these oncosomes have all 24 chromosomal intact DNA, whereas exosomes don't. And that's why you cannot see them under the light microscope. And in the literature, you will see the word cargo. For better or worse, it is stuck in the literature. What they're referring to is the content inside the exosomes and oncosomes, whether it is RNA, protein, and a little bit of DNA in the exosomes, but entire DNA in the oncosomes. And the key feature here is exosomes are produced by all cells. We need the exosomes for our lively biological processes, whereas oncosomes are only produced by cancer cells. And relatively, it is easy to isolate the oncosomes and characterize them. And that's what we have done. Next slide, please. So let's go into some literature evidence. And this paper says large oncosomes contain distinct protein cargo and represent separate functional class of tumor-derived extracellular vesicles. And next slide will show on the right side, next slide, please. On the right side, you will see the distinct cargo from oncosomes versus exosomes. And by now, you know that by centrifuging at 10,000, you get the oncosomes, and centrifuging at 100,000 with ultracentrifuges, you get the smaller exosomes, and the cargo content is totally different. On the left is a confocal microscopy showing the highlighted box, the budding of the oncosome that is stemming from the cell membrane, and therefore the green antibody staining that you see on the tumor cell is extended to the bubble, and therefore the oncosome it can be really considered as a daughter cell, cell-like organelle coming from the tumor cell. Next slide, please. This is a prostate cancer publication. It says large extracellular vesicles, they're referring to the oncosomes, carry most of the tumor DNA circulating in the plasma. And so the results show that most extracellular DNA is packaged into the large exosomes as opposed to the, oncosomes as opposed to the exosomes. They are chromatinized. I have already showed that we have proven that all 24 chromosomes are there, and they mirror the genetic makeup, and also they have specific tumor abnormalities, which I will show you. Even though this is a prostate publication, this theme continues for all solid cancers. Next slide, please. So let's take a look at the advantages of oncosomes. They are abundant in plasma compared to the tumor cell. We always start with 3 cc's of blood. We'll be lucky if we get few cells, tumor cells, but it was stated in the literature that each tumor cell is capable of producing up to 2,000 oncosomes. Therefore, it's easy to study them and easy to isolate them. And as I said, only produced by the tumor cells. and they represent a snapshot. When you study a patient at a given time, since the half-life is short, and you can study what is happening at that particular point in time. And as I stated, they have the similar genetic profile, and more importantly, oncogenic messages are enriched from the tumor cell and pumped into the oncosomes. Next slide, please. So here I'm showing you a parental tumor cell and an oncosome, and you might be wondering, I told you oncosomes are small, how come it is bigger? It's a blown up version. But when I make an informal presentation of this, I seek the audience and say, can you count the gold spots on the left? And they always say 9. And then on the right, you have 9 to illustrate that as far as chromosomal content, the parental tumor cells and oncosomes have the similar genetic feature. Next slide will show you specific gene signatures from the tumor cell and the oncosome. And I'm sure everybody in this audience is familiar with HER2 amplification. When it comes to the breast cancer, FDA insists that you do the genetic test before you start the Herceptin treatment, because if the tumor is HER2 negative, patient not only will not get the benefit of the treatment, but there are side risks and etc. So what I'm showing is a cell line, SKBR3, breast cancer cell line, 3 cells showing amplification of this particular gene, and you see multiple dots and also big patches there. And that big patch is the really amplification of 100 or 1,000 copies. And then when you move to the right side, it is an oncosome also showing a similar pattern, small patches, and also the big patch. So I have shown you that the tumor cells have the similar genetic composition in general, and specifically the genes that are oncogenic for the cancer process. Next slide, please. So in our proof of concept study, the design obviously by design is non-blind. And when we receive these samples, we know which one is normal and which one is tumor. And roughly there are 100 subjects involved in the proof of concept, which is what I'm going to show you the results. Roughly 50 normals and 50 cancer patients. And the control subjects obviously came from family, friends, and non-cancer patients from the largely 2 physician offices. And so the patients comprised of active cancers, history of cancer, or they were in remission or in relapse. And as I said, these samples came mostly from 2 locations, one from Orlando and one from Alabama. Next slide, please. This is showing the results, and each dot represent one patient result, and the x-axis is the total number of patients. The y-axis is the oncosome count, and I hope everybody can see at the bottom the red line, which is a cutoff. So everything below that red line represents a normal tumor burden, and it is at 12 oncosome count, and everything above 12 represents a tumor burden. And most of these dots are proven cancer patients. There are a few exceptions that I will highlight in the next slide. So here, for clarity and for meta-analysis, I removed all the confirmed cancer cases above that red line. And similarly, I removed all the normal adults that don't have the cancer from a clinical standpoint. So what is left below that red line are the patients that actually went through all types of treatment. These represent all kinds of cancers. And when they successfully finish their treatment or very close to their treatment regimen, they were clinically, you as a clinician declared that these were in clinical remission. And when we look at the genetic remission, number of oncosomes, there is 100% concordance between the clinical remission and the genetic remission. So if you go far right and look at this circle, there are 4 patients here that are on treatment, but they have, at the time of this study, this was about 2 months ago, they have not achieved the complete remission from a clinical standpoint. And similarly, they have not achieved the genetic remission. So the assay is also consistent in that respect. And then if you go to the far left, there are 3 clusters here, and these are in the box. And mostly 2 clusters. There is an outlier at the far left. And these cases do have, as you can see, above the red line, the oncosome count above the 12. So traditionally, you would call in an assay like this false positive. I have already been warned to change the term, which I already did, and we started calling them presumptive positives. There is a reason why, and I will show you a couple of examples of cases where they actually turned out to have the cancer. Next slide, please. So let me show you some cases where I will illustrate the point that as I'm talking to you, the assay is perfectly suited for monitoring the disease, regardless of how you detected it. Once you put the patient on treatment, the assay is tracking the disease. And here is a patient with colon cancer. We didn't get the opportunity to study pre-surgery. We just estimated the number, but we had the opportunity to study 3 times. And then, as you can see, the oncosome count went down, and around March 23rd, the patient was declared to be in clinical remission. And then next study was also clinical as well as genetic remission. Next slide, please. I want to share a patient that all of you can recognize that there seems to be a rash-like and in the earlobe. And next slide. The biopsy showed that this is a basal cell carcinoma on April 20th. The surgery was done around end of July. And the next slide, please. So at the time of the study, surgery, just before the surgery, we did the, I apologize, this is a little bit fuzzy, but I'll walk you through. The top left is a study 1 month after the surgery, and this patient happened to have the highest number of oncosomes in the entire study, about 600. I'm only showing you just a snapshot of one field. And also, there were 600 before surgery, but that was only a few weeks, 3 weeks after the surgery, roughly 1 month. Then if you go to the top right, it is 2 months, and the count went down to 25. And again, it is a representative field. And then 3 months later, the count down to 4, which is in the normal range. So regardless of the type of the tumor, we are finding that the body is flushing out these oncosomes when the patient is on treatment, just around 8 to 12 week time period. Next slide, please. And that is showing graphically the demonstration of oncosomes going down and agreeing with the clinical situation. Next slide. And this is a very interesting patient, very timely for this presentation. This is a patient with colon cancer with a liver metastasis. And you can see at the top right, there was colectomy done almost a year ago. And then you can follow the CEA levels before surgery was 40 and was going down almost to the normal range. I was told 3 to 4 is about the normal range. By the time we had the opportunity to, there was a liver mass ablation in July 15th. And by the time we had the opportunity to study the patient in October 14th, when the CEA levels were just about the upper normal range, we had 0 oncosome count. And we would predict that if we had the opportunity to study before, they would have been higher based on the other experience. And 3 days ago, the husband of the patient called me. He himself is a physician himself. Told me to do this study, and I immediately asked him to send the blood. Yesterday morning, 8:45, we received the blood sample. Exactly 10:45, I had the results, and you can see that the oncosome count is 29, and it is clearly tracking the disease and going with the established CEA marker. So we feel very, very strong. And then I have shown you cases where it is going down. And this one is clearly showing you in October, there was nothing. And in December 2nd, yesterday, there were 29. So this is the first illustration where when the patient is coming back relapse, this assay can detect them at a very, very early stage. Next slide, please. So I wanted to show you this interesting case, even though no firm diagnosis of cancer was established for this case. It's interesting because take a look at the size of I'll show you the image in the next slide. Take a look at the size of the nodule and how the marker is correlating and tracking. The middle one, we just estimated it. Next slide will show you the imaging of this. I'm not a physician, but I'm assuming the lower left is where the mass is showing about 1.5 centimeters. Next slide will show the diagnosis around May. And next slide, please. This was at 1.5 centimeter, and now it is at 0.4 centimeters. And the next slide will show you the date, July. So I wanted to share this very interesting case where a reduction of a nodule, which the exam did say that it is speculated. In general, as all of you know, speculated nodules generally correlate with the cancer, but no definite diagnosis was made in this case. But I wanted to share this interesting case. Next slide, please. Now I want to turn to the presumptive. Okay.
Please go ahead.
Okay, thank you. So now I want to turn to the presumptive cases, why we feel this is a sensitive screening technique and it is picking up very, very early. I will start with the case on the right because the entire study started on January 3rd of this year. I was in the physician's office, the co-investigator of this study. He examined the patient. She came with a complaint in the chest. He examined the breast, could not feel any lump, clinically declared normal. And I was in the waiting room, and the patient came out and said, "Doc, I'll be happy to be the guinea pig," because she heard about this study. We drew the blood. I went to the lab, and 2 hours later, I saw 69 or 73 oncosomes from 3 cc's of blood. And I called the co-investigator and said, "I see the evidence of cancer." I didn't say breast cancer. I said evidence of cancer, because using this screening assay at that stage, you can only answer the question yes or no. And as you see, 5.5 weeks later, there was pathological confirmation of invasive breast cancer. And now switching to the left, this is a friend of one of our, oh, can you go back? Yeah, thank you. This is a friend of our investor. I talked to him and he said, "I'm very active, I work out, and I feel very good, I'm in good health. But lately, to paraphrase his words, I'm out of the game and I'm feeling down, etc." And I said, "Let's take a look at your blood." And we got the blood sample. August 3rd, and we saw 30 oncosomes. And then at the end of the August, he was diagnosed with the cancer of the throat and inside. So this is considered pathological conformation of the tongue and the throat. That was around 6 weeks. So see the two cases that since the half-life of these oncosomes is short, when we do see the tumor burden, it is indicating either the presence of the cancer at that time or it's about to advance. So I wanted to share with these two cases out of those presumptive already turned out to be positive, and therefore let me switch to the definitive diagnostic side. As I speak right now, we can answer the question when there is oncosomes. Is there a cancer in the blood? Yes or no? And obviously, as a clinician and also the patient, what is the first question you're going to ask me when I say there is a cancer? Where is the cancer coming from? What type of cancer it is? Right now, I can tell you yes or no. I can also tell you whether it is coming from hematologic lineage or the solid tumor lineage. And what is shown here is a profiling of the cell membrane. And if you recall, this cell membrane is actually coming from the tumor cell. Remember, this is like a daughter cell organelle. So using the proprietary technologies, we can tell you if there is only red stain, this is a solid cancer. If it is red and green, it's a hematologic malignancy. But going one step further, under the hematologic malignancy, we do have a case. The gold stain shows that it is multiple myeloma or plasma cell, and the blue stain shows that it is a kidney cancer. So today, I can only give you definitive diagnosis of the one cancer type under each lineage. But if you recall, let's take this oncosome. The profiling is outside. But I told you inside, there is a cargo that has the DNA, RNA, protein, and we're going after the specific microRNA. As I'm talking to you, the research is already underway, and we're going to use the same oncosomes, go inside and profile them. We have already identified individual specific microRNAs, and hopefully within 2 months, I'll be able to come back and share with you that we now have the assay that will answer the 3 questions. Is there a cancer or not? What lineage it is, and exactly what cancer type it is. So stay tuned. So here is a summary of the proof of concept. I have already addressed the red 10. I showed you presumptive, and we would like to see no false negatives. But in this small study, there is one false negative, and this patient had mastectomy, breast cancer. We did 2 times prior to surgery, 1 time 0 oncosomes, and the next time 1 oncosome. So we don't know what is happening, but hopefully in the larger study, which is what we are here to seek your cooperation and guidance, hopefully we will stick to very, 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 very low false negatives, and then also improve the presumptive. So we don't have time to go through all the mathematics, but if you just go back to the previous slide, I wanted to point out that in any assay that we develop, sensitivity, specificity, and accuracy are important, but look at the 100% accuracy in terms of monitoring. Next slide, please. So I came up with some guidelines for the moment. For monitoring purposes, hopefully I have shown you that this is a robust assay. But if you have clinical suspicion and happens to do this assay, if the count is 13 to 24, follow them up every 3 months. If the count is 25 to 49, increase your surveillance. And if the count is 50 or above, do everything you can using the gold standards to find that nodule. It is there somewhere. But if you recall the patient that we just did yesterday, her count was 29. She had an MRI of the liver, and they're going to do the PET scan CT, I guess, next week, and we'll see what the results are. But for the confirmed cases, if possible, obtain a pre-treatment oncosome so it will establish the baseline. And then when you follow them up, if the count is trending down, you may either reduce the number of the invasive procedures, or you may, at some point, use this non-invasive procedure totally. So our results of the blind study will confirm that. But this is all in the interim, when I cannot give you the definitive diagnosis. But 2 months from now, even when clinically nothing is remarkable, if I see a signal coming from, I'll be able to guide all of you clinicians that concentrate on this organ. The message is coming from this, so try to find the lesion in that organ. Next slide, please. So what is the purpose of our meeting today? Again, I apologize for the technical nuances initially. So we are seeking your help to do the blind study to confirm our proof of concept results. And I have done double blind studies all my life, and I coined the term triple blind. I don't know if it exists in the literature or not. There are 3 spots in this triangle: me, principal investigator, you as a participating investigator, and there is a nurse coordinator. So the way this blind study will flow through, there is an alphanumeric code around 600 already generated. The phlebotomist will draw the 3 cc blood from EDTA. You will have all those guidelines, and send it to the laboratory, and you will keep the paperwork with informed consent and send the clinical data to the nurse coordinator. And when we have the results, we will send the results to the nurse coordinator. So all of this information, the common link is that alphanumeric code. So when the study is complete, hopefully, I want to finish this by Christmas, by end of the year. We want to recruit 300 cancer patients. Please include 10-to-1 ratio, some normals, 10 cancers and 1 normal. And the next slide, please. So what are the study materials? You will have a copy of the IRB approved, central IRB approved protocol, informed consent in English and Spanish, the labels that I already talked about, the blood drawing instructions, our FedEx and UPS information you can use, the study coordinator information, so that if you have any questions clinically, call her. If you have any questions administratively, call my administrative assistant. Next slide, please. So what are the inclusion and exclusion criteria? We have, you can include any patient at any stage of the treatment, either prior or during, in remission or relapse. You can include children of all ages, including the pregnant woman. IRB said CLL patients will not get benefit out of this study. You will include them. Most of the CLL patients are indolent. They're not even on treatment anyway, so exclude them. So you cannot include CLL patients. Last slide, please. A study like this would not be complete without, for lack of a better word, an army of colleagues. I want to sincerely thank my co-investigators, Dr. Koneru, who is hosting this meeting, also Dr. Singh from Orlando, and then Dr. Cherukuri, hopefully he's in the audience, from NIH, who is a constant inspiration for me and for his strategic help. And my administrative assistant, Banal, who you will call for all clerical help, administrative help, and the nurse coordinator, Ro Weiss, you will call for any clinical help. And all the technical work is done by Mr. and Mrs. Fontes, my lab associates here. And Shri Kopuri also did some studies, but helped me with the presentation, preparing slides, etc. But there is one individual I want to thank. Without her help and putting up with me with all my crazy schedules and constant encouragement, without my wife's help, nothing would have been possible. So I really thank her. With that, one more time, apologies. I'm not lying to you. I have given hundreds of presentations, and a technical goof up like this happened once. and this is the second time. So I apologize, and I will stop right there and entertain any questions.
Thank you, Dr. Babu, and Dr. Satish is here. Dr. Katula, please take over.
So can you unmute me on my phone also, please?
Okay, is it Dr. K's phone? Yes, I think that's what it is. Dr. K phone, is that right?
Yeah. Can you hear me okay?
Yeah, we can hear you.
Okay. Thank you so much, Dr. Babu, for that presentation. We do have a question from audience. Dr. Jayshree Jagadhar, have you worked with Emory University? I would like to work on lung cancer. We have plasma frozen tissue. Can you use these to detect oncosomes?
Okay, excellent question. Right to the target of plasma. The oncosomes are present in plasma, as I showed you. And we have done extensive literature search about the stability of these oncosomes, and especially the cargo inside the oncosomes. And if these are frozen and in an academic institution like that, I'm pretty sure the protocols are very robust. And I feel very, very strongly that the frozen oncosomes are very useful for the study. And in fact, the way we are doing our clinical study, we are dividing the samples into 3 portions. The first 1/3 will go for the enumeration of the oncosomes, as I showed you. The second third will go for the antibody studies to show you the profiling of the membrane. And we are freezing, just like you did, the oncosomes, the third portion, but we were freezing the oncosomes after we separated them from plasma. But I don't think there would be any problem. We could do just a couple of things to make sure, and then I'm pretty sure the plasma is frozen plasma will be very useful.
So another question from Dr. Santosh Kesari, how does it compare to Guardant or Gallery tests that are commercially available right now?
Okay, another excellent question. So let me take through some scientific medical aspects and some operational aspects. This is a study that claims, again, from liquid biopsy, 50 plus cancers. And I have read their data. And to the best of my knowledge, the results are given in percentages, that when you have certain messages, they do the sequencing, and they give probabilities of you have 56% probability of this cancer or that cancer. And so to my knowledge, this is not useful for detecting early stage. However, most of their data seem to be pretty robust in picking up the signal for the stage 3 and stage 4. But everybody, the holy grail, and I have been hearing all my life, is early detection, early detection, early detection. Everybody talks about early detection, but I haven't seen much in practice of doing it, simply because technology is everything. There hasn't been enough evidence until now, hopefully, to detect early. And as I pointed out, we're detecting way early that the gold standard techniques are not picking up. And in my opinion, using the layman terms, when we see an oncosome count, I say it's a clicking time bomb. It's only a matter of time that the patient will or might convert into the frank cancer. But in this respect, I want to bring one study that claimed using other genetic technology that you can detect the cancer 4 years before it actually developed. So coming back to Galleri test, in fact, we did send one of our patients, and now I'm turning to the operating aspects. We did send one of our patients a sample to Galleri. They claim about 4 to 7 day turnaround time. 30 days later, we haven't received the results. We called them, we finally got it. And it is, it can only be done in one location in California. As I told you, ours will be a point of care test. Everybody in this audience, I guarantee you, I can train you in half an hour. You will be able to do this test. And most importantly, cost effective. This is over $1,000, and ours will be relatively inexpensive compared to that. So both on the technical professional aspect of the assay versus operational assays, there are significant differences. And I'm aware of only one other test, maybe from UK, that also claims, but I don't have much information, but these are the only two tests that seem to claim that they can detect. Hope that will that has answered your question. The take-home message is, my opinion, at an early stage disease, that technology is not sensitive enough to pick up.
Okay. So another question from Dr. Shipra Garg, what molecular technique is applied to detect the oncosomes in blood? What kind of molecular technique?
Okay, very excellent question. To start off, let's go through the sequence. When we do the initial, first of all, we have done extensive molecular profiling, the cargo, DNA profiling of the oncosomes, and have proven, taking from 9 cell lines and 9 cancer cell lines, different cancers, and 1 normal lymphoblastoid cell line. And we were able to show that oncosomes were only produced by the cancer cell lines, and no oncosomes were produced from the normal cell line. And then when we did the profiling of the tumor cells and the corresponding oncosomes, they are very similar. So the molecular technology that we used is a molecular cytogenetics, DNA fish probes using fluorescent tag. We were able to show the genetic makeup is similar, the tumor and the oncosome. Then it's a little bit involved study. I told you that every one of you in this audience will be able to do the study, but if you have to do that profiling, you will not be able to. You have to depend on a geneticist. So then I turned to the DNA stain, which is all the 24 chromosomes inside the oncosome. If you use the DAPI stain, you can highlight them, which I already showed you the oncosomes. So for the initial enumeration, to answer the question, is there a cancer or not, you don't have to go for the molecular technique. Just a simple stain will answer you. The second step to go for the lineage, it's an antigen antibody cell surface that I showed you those color, red, green, etc. Then the diagnostic test that we are doing directly relate to your question of what molecular techniques we're using. We are using one type of RNA. There are multiple types of RNAs there, but the microRNA is a very, very unique species of RNA. It's a non-coding RNA. It doesn't produce the protein, but it is a highly important RNA that regulates the tumor genes and all of those. And therefore, we have identified specific microRNA. So we'll be using the profiling with a modified modified amplification technology, not PCR, but it is called rolling cycle amplification, RCA. And that can also be done at an isothermal, meaning right on the laptop, benchtop, and no separate instruments are needed. So we will use that to amplify the oncogenic signals, but then the detection is back to the fluorescence. Therefore, we are combining the two molecular techniques to come up with a diagnostic assay. So the answer is all the technologies that we use for the specific diagnosis are molecular. Hopefully that will answer your question.
So can this be done with saliva?
Ah, okay. This is another interesting question. The oncosomes have been shown to exist in various body fluids and obviously saliva. And I have worked with saliva for a different reason. And I think you probably will agree, we're not out of the woods yet as far as COVID is concerned. And I came up with an assay from saliva for COVID that will detect in 15 seconds. So there's a lot of interest for medical use of saliva for other analytes. And I'm aware that oncosomes have been claimed to be present in other body fluids, including saliva, but we have not tested the saliva for this, but it is probably feasible to use it, but I have not done it personally.
So Dr. Satish.
Another question from Dr. Sharda Gupta. Do you recommend this test for the family members who have cancer history in the family?
Okay, the first thing that comes to my mind is obviously the colon cancer, the genetic disposition, and also the BRCA assays that will show family members whether they're at an increased risk or not. But remember, this is not an assay for predisposition. This is an assay that detects the actual presence of the cancer in a snapshot. So therefore, if there is a family history of cancer, and it is well known in the medical community that if you have a history compared to the families with non-history, there is a slight increase. Slight is a relative term. So not only the family, yes, definitely we will recommend doing this screening assay, but the ultimate goal is to use this when the patients come to all of your offices. I'm sure there are primary care physicians also in this audience, and there is a big checklist annually, metabolic profile, this and that. We are hoping that you will add another checkbox and say, check my oncosomes for annual screening, and then go based on the findings. So yes, definitely we will recommend for members of families with history. But remember, for the most part, with the exception of few cancers, I already mentioned breast and colon, most of these cancers are sporadic. These are not familial. So having said that, when we are recommending an annual screen for general population, certainly it goes without saying that family members with a history are justified to go through this screening.
So
we have several other questions, but everybody kind of back patiently, even though there was delay, which is which shows how excellent your presentation was, and your questions, answers are very interesting, and definitely this is something that to look forward to. I'm an oncologist, and I see a lot of patients, and we monitor patients. We don't see anything when we do the CAT scans, but again, at molecular level, there are cells floating around. So this certainly will help us in future. But how we are going to really use extended treatments remains to be seen, because still this is not prime time. Again, studies have to be done with this data. This can be used as a tumor marker like CEA or PSA or something like that, but this can be much more sensitive if we have a baseline oncosome study, oncosome levels. But there are a few more questions. Maybe we can do a couple more questions, and then we can stop, because it's been a while since we have been doing it. So let me see. There is a question.
Satish, can you hear me, Satish?
Was that Dr. Padma Narasimhan, who is an oncologist from California? So she has a question. Garden test is expensive, but is covered by insurance. This is not. So how much does it cost the patient? I don't think we have an answer, but yeah, please, Dr. Mesh Babu. Can you answer that question when it is available?
Initially, this may be a fee for service like any other assay. But as I said, I wanted to make sure that all the participating physicians is a request from my side that I showed you the proof of concept, but we want to finish the triple blind study. We want to include 300 cancer patients with, if you see 10-to-1 ratio, maybe around 330, and we already have identified sources to include 100 additional normals. So we feel that the insurance companies, the dialogue has already started, that they would very much encourage, because of the clear expectation of cost savings, that if you can detect much earlier with this or any other methodology, that will be overall good from a cost standpoint. So we haven't finalized what the, if you will, the retail price is at the laboratory level, but I can tell you certainly it will be a reasonable assay when you compare with competing technologies. So, I'm sorry I can't give you the final figure, but it will be a few $100s.
Right. We have Dr. Murali Cherukuri joining from NCI, NIH. He's a senior investigator, and he has no financial involvement with this company, and he has only the academic interest in this line of research. And I just want to clarify that.
So one last question from Dr. Amit Chakravarti. He's asking, can I send my blood for testing? Any discount for AAPI members?
In fact, this particular point, as I said, the proof of concept study, I mean, technically ended, but we are about to start the triple blind study. And Dr. Koneru brought this to my attention that if a few members are very much interested in participating, I said, why not? We will open, as long as it's just a few samples. We can include them in the non-blind proof of concept. And the reason is you are so much interested in knowing it. And if you send your blood and it is part of the blind study, unfortunately, I cannot give you and share the results, and because I don't even know which sample is yours until this study is complete. So my recommendation is, of course, we will forget about the discount and all that. We will do it for free. We will include that in the proof of concept continuation. And sure, yes, we can do that.
Yeah, if anyone is in Alabama, Huntsville area, you please call me. I can arrange for that through my office. Since this question came from Amit, we can discuss after the meeting.
So sorry we can't get to all the questions, and now it's 12:21. So thank you so much, Dr. Ramesh Babu, for that excellent presentation.
I have one request, and I know it is recorded. If you can send the transcript of all those questions, if it is possible, we will answer every single one of them. And also, I will provide to AAPI it is only a 1 minute video demonstration of our technique, and it is unfortunate that I could not send it through the email because it's a huge video file, but I will provide that to Dr. Koneru, and then the organization will have an opportunity. Maybe you can post that link and then you can watch that video.
No,
Thank you, Dr. Babu, for that. So appreciate that. So thank you, Dr. Koneru, for hosting today's meeting, and thanks everyone for joining.
Thank you, Dr. Satish. So there was a question about the contact information for Dr. Babu. I'm going to put it in the chat box, and if you can copy it and paste. And
And also for everybody that is interested in participating, there is a package that Dr. Conero already has, including contact information, as well as the IRB approved protocols and all that.
also, Dr. Ravi Kuli is online. So,
Yeah, hi. Thank you so much, Ramesh Garu, for an excellent presentation. I could not join as I was flying into Chicago for another local chapter meeting. It seemed like I joined at the end. The questions and your responses seem very captivating for me. I'm going to listen to the recording, and I would like all our AAPI members to be part of this cutting edge technology to kind of advance the science as well as the patient care. I think it will help a lot to be in the forefront of this technology implementation and adaptation. Thank you, Chandra, for bringing this topic to our attention. Thank you, Ramesh Babu. Thank you, Satish, for taking care of the program today. Have a good day.
All right, thank you very much. Thank you very much, Dr. Kohli, for giving me the opportunity.
My pleasure.
thank you all for joining. And if you have any questions, please contact Dr. Ramesh Babu or myself or Dr. Singh. And as I said, I posted those 3 contact numbers and email addresses in our chat box. Again, once again, thank you all for joining, and we'll see you all soon.
All right, thank you.